affectionately called IndiaAlliance. What is IndiaAlliance? We are an organization that was established 10 years ago with funding from the Wellcome Trust, which is a British charity, and Government of India through the Department of Biotechnology. We complete our 10 years, and to celebrate this 10th birthday, we are starting with something that's very dear to us, and that is public engagement, communicating science to the public. We hope that you young college students who are with us today will be our fellows of tomorrow. The series that we are starting today with these public talks is called the Explorer series. And 4 of our fellows would be giving talks on their work. Why the Explorer series? Because every child is an explorer. Every child is a scientist. Education is meant to enhance that curiosity, not kill it. How many of you have, as kids, tried to do something and you were told, don't do that, you'll break it? Take the example of an egg. A broken egg is a physics experiment, an experiment in gravity, an experiment in how fluids move. It's also a destroyed biology experiment. In your kits, you have these pencils that have seeds in them. We hope that you will use that to grow a plant and do a biology experiment. So welcome to the Explorer series. We're very glad you're here. Thank you for being here.
Good morning, everyone. Thank you, Dr. Jamil, for introducing the Explorer series. So first up, we have Dr. Farah Ishtiaq. She's an ecologist who's fascinated by avian malaria. And if you've not heard of this field of study, that is because not a lot of people in India are in this field. But Farah intends to change this, and we'll talk to you about why it is important that we worry about birds getting high fever and blood disorders. Trained in avian ecology and behavior from Aligarh Muslim University, Farah is committed to conservation and has been instrumental in bringing together the Indian Bird Conservation Network. So over to you, Farah.
Thank you. I just speak in this.
You can use this one.
Okay, fine, that's fine. So just let me get this right, which is forward. Which is the pointer? Full screen mode. Is on here, yeah?
Yes.
Which is the pointer here?
See, this one is the pointer.
Okay. And then forward I can do from here. Right, okay, that's fine. And this is, oh, yes, fine, thank you. Good morning, everyone. I've been dreaming about this for a long time, that I'll have an opportunity. So I feel really honored and privileged to be invited. You know, there are a lot of fellows we have in IndiaAlliance, so we'll also have to compete who can be invited to give a talk. So I feel really honored. And I feel even more honored and excited when I received this funding, which was 6 years back. So there have been several challenges I have faced in my career where I felt, things may not work, and maybe this might be the end of my dreams, where I want to continue in science, working on birds. And malaria is just an excuse. I work on birds. That's why I want to make it clear. I really like working on birds, but I tell you why. So this is especially for all the young women sitting here. Please, men, don't mind. Please be persistent and determined what you're trying to achieve. Don't let your dreams die. That's the only message I'll give you today, okay? If you keep it alive, it will work. Nothing can stop you from achieving that. So I work on birds in malaria, and I've done my PhD on birds in ecology and behavior, and that was something like 12 years ago when I was just watching birds, looking for their nesting behavior, ecology, how they interact with the environment. I suddenly realized that, you know what, it will be really nice if I can bring some molecular or genetic aspect in my study, because there are some answers which are actually hiding in their DNA. And if somehow I can combine that with their ecology, that might actually bring a new perspective to my science. So I've been looking for a postdoc that time, and my first postdoc, which I embarked on, was in Smithsonian on malaria. That's what I offered. Now, beggars cannot be choosers, right? If that's the opportunity you're getting, I took it on board, and that's the most fascinating thing I've done in my life that time, and it opened a new whole world of parasites and diseases in birds, and I could never look back since then. So let's just think about malaria from human perspective first. I would like you to think. So you know that malaria kills a lot of people, I think more than 600,000 or 700,000. There are different reports. You look at the WHO report. And mosquitoes actually are responsible for transmitting malaria in humans. So even though we have, say, something like 500 species of mosquito, there is a very minuscule amount of actually mosquito species which can transmit these parasites, which is Plasmodium in human case. Now, the things get even slightly more complicated. There are actually 4 main things you need to understand about malaria to persist in a population, which is first, you need to have a host, which has to be a competent host, which means somebody who is not very has to be immunologically challenged, a weak immune system. You need to have a very competent vectors as well. So in case, if you have Anopheles mosquito, which can transmit a Plasmodium parasite, then it will persist in a population. And then you have Plasmodium, which has to be in the population. But even if you have a host, parasite, and a vector, does not mean the malaria will transmit. You have to have right environment as well for this parasite to transmit. And this is a nerdy slide I have put here, which just shows you the development of malaria. So there are a lot of models which people have proposed that it goes up as you increase the temperature. But it's not a linear relationship. It sort of drops down, because beyond a certain temperature, transmission cannot take place. You know, the parasite will not develop. So which means there is a temperature threshold for these parasites. So if it becomes too cold or too hot, transmission will not take place. Same in birds. The story is the same. You have different kinds of malarial parasites. In fact, the diversity is much higher than what you have in humans. So in humans, you have 5 different parasites for human malaria, but birds, you have even hundreds of them. It's all about the host switching. They shift their host and everything. And again, the transmission is also very much related to the temperature condition. If everything is so temperature related, then climate change becomes a bigger issue here, right? So when there will be increase in temperature, as we all hear, there will be a shift in the transmission of these parasites. So there are areas which are not conducive for parasite transmission. Tomorrow they will become very good for some other places. The parasite may even completely become wiped out for non-suitable conditions. Now, from my perspective, from a bird ecologist perspective, I feel that birds are a better model to understand this, because in case of human malaria, you have vector control, socioeconomic issues, you also have infrastructure, insecticides issue, which can actually dilute this effect of climate change, which somehow in the wildlife diseases the effect is not so strong. So it's easy to sort of tease apart some of these issues there and to understand, is it really going to be influenced by the change in temperature? Now for the birds, the story gets slightly complicated here. So in humans, you have only Plasmodium parasites. In birds, you have a Hemoproteus as well, which is again a blood parasite, behaves exactly like a malaria parasite, and then something called leukocytosome. And they all have their own vectors. So if you know what your population has in birds, you can tell which vector might be playing a role here, just looking at the prevalence of that parasite in the population, basically. And And from ecologist perspective, when we talk about how the birds are declining and extinctions are taking place, even more ecologists also have been blaming just the population growth or habitat destruction, pollution, effect of veterinary drug in some cases have brought down the population very low in some species. But actually, disease ecology has never been considered as one of the factors, which actually has become very important, have been important from the, what happened here? From the climate change perspective. And if I talk about the negative effects of malaria, yes, like in humans, we get malaria, we get anemic. It just, lowers the blood cell counts. If you have low immunity, it also compromises your immune response. It has effect on your fitness. So you must have heard of this hypothesis that birds select good genes hypothesis. They select mates based on their good genes, so how brightly colored or beautiful they are. So the ones which are more parasitized, they never get selected. So it does affect the influence, right? They can't sire or they can breed in the next generation. And the reproductive success. There are studies in Europe which shows that here there is a high prevalence of malaria parasite. There was a very less, the birds which have bred or they didn't have any broods. And finally, in very extreme cases, use the mortality. The birds do die. This is the work I did for my postdoc, which I found very fascinating, and I'll go over just for a few seconds. So this is about Hawaii Island. So just imagine very nice, pristine Pacific islands. They are so far off US coast. And the birds here on these islands, most of the species are endemic. They have evolved without any blood parasites or any pathogen. So their immune system is particularly different from the mainland birds who have been exposed to different parasites. Now what? Humans came, they arrived. They brought their own pigs and they started doing agriculture. They bring their poultry. That didn't have any effect until the Europeans arrived and they brought not just all the trade, they introduced a lot of birds from the mainland because they felt bored of seeing those endemic birds. They wanted to feel at home, so they introduced a lot of species from different parts of the world to make it look more vibrant. And guess what happened? Accidentally, with the trade and all, this species of mosquito, which is a very common species all around the globe, was also introduced. Birds brought parasite, mosquito arrived, good match, very nice, good temperature, and transmission of parasites to endemic birds, which have no immunity to parasites. So what? Extinctions takes place. Birds started to die. And so far, there is only one species of mosquito and one parasite which persists in Hawaii. And that's what we've been looking into. Now, we managed to sort it out, that where they got these birds from and how the structure evolved. But you might be thinking that, so what? It happened there. We are sitting in India where we live with these parasites. We have human malaria so prevalent, so why we should worry about it? I'll tell you why. Because we also have a very fantastic migration system. So there are a lot of birds in Himalayas which migrate. But a lot of birds don't, which live in very high elevation environment, 3,000 meters. They just breed. But some of them, they do come down in the plains in the lower elevation every year as part of the migration, and then take these parasites back. So if you have, so supposing this is the migration thing, so this is your breeding ground in high elevation, and this is the wintering ground. The birds are coming down in winters where they're getting exposed to all sorts of parasites or vectors, and then they go back to breed again. Now, this is a good question in terms of migration ecology. Why do you want to go back if you are there and if you're getting all the food and everything nice? Because they also want to escape parasites. This area is quite infested with parasites. The conditions in the winters are too harsh, too cold, and the food resources are much nicer in the lower grounds. So they migrate back. But in that process, there is something called migration culling. The ones which get most infected, they never make it back to the high elevation. So it's like a weeding out the ones which are most infected. However, the ones which have made it far, they probably have very low intensity parasite or none at all. They occupy the breeding grounds and they breed. However, if you have the right vector species in the high elevation, which we didn't know whether there can be transmissions, and just like Hawaii, the birds which don't migrate in high elevation, they can get infected with these birds and can lead to the mortality. Now, this process has evolved over the years, like millions of years. But the climate is changing now, right? We don't know whether the birds are being exposed, whether there has been any change in the population dynamics with relation to disease or the birds. And that's what we are trying to tease apart, that whether there is malaria in high elevation environment. So to look into that, I've been sampling these birds for the last nearly 8 years now. I have 7 sites in Himalayas. Anyone interested, please come contact me. I'll be very happy to take some of you, not all of you, if you want to join me in the field. We do mist netting, like we catch the birds, we sample them for blood. You put a metal ring, which you can identify them. If you retrap them, that's like a unique identification number. Oh, it keeps going off. And then you can basically make slide smears, and then you can do a genetic analysis of the DNA as well. And apart from that, we have a huge selection of species. We know how far they move, how far they go across during the elevation. And you have a nice selection of species. You can test several hypotheses to understand that whether the malaria is being transmitted only across the elevational migrants, or is it within the resident species as well. Now, the vectors are an important part. As I said, without vectors, this transmission cannot take place. So we have been trying to understand how some of the vectors are found, say, across the elevation. As you can see, it goes down. As you go up in the high elevation, there is hardly any vector species. There's like a peak abundance here in around the June or July or August time. But the birds are breeding somewhere here, so they're sort of like escaping these vectors. I can't say more than about it because I need to think more. I've just figured out this data in the last few weeks, but it's very exciting information. So a collection of all this information actually helps us for other diseases as well. So we've been hearing about Zika cases in the last few years. But I feel in terms of the disease ecology, we have not done much, actually, because everybody likes to see what strain of virus is there. But actually, if you collect fine-scale data on the vector dynamics and everything, you can actually do a predictive modeling and to understand when and how these outbreaks will happen, and can we do something about it. So that's something I want to say, and I'll open the room if you have questions and something to discuss. Over to you all. Please ask questions. I'll be very happy to. I deliberately kept my talk short. Not that I ran out of stuff to say. Come on. Yes.
Some of these parasites that infect birds, is there anything more they have crossed the species and infected humans, like we know for Plasmodium nonzae that was infecting?
Okay, so mean cross species. So, right. So the question was that do they have cross species infections like Plasmodium nonzae? You think is transmitted from apes to humans, basically. So the thing about avian malaria, it cannot cross-infect humans. So there is no chance of that. My research will be very important if that ever happens, actually. But within the birds, they do switch host. So like a common manna, if it's infected with a Plasmodium parasite, the other, say, parakeet or a sparrow can also be infected with that. So but it's also, there's some sort of generalist parasites, and there are some specialist parasites as well. Yeah. Yes?
I'm a clinician, so obviously bird flu. So I'm wondering about the interaction between your wild birds and the domestic birds. So have you looked at that?
What do you mean by domestic birds?
Domestic chickens, ducks, which are actually reared, which are actually in herds.
Right. So a lot of, I think it depends in what habitat they are using. So like, of course, the water birds, they do tend to, we do tend to see a pattern. The birds sharing a similar kind of habitat, they do get infected with similar sort of parasites, but it's always not as clear. Domestic chickens never get. They have their own gallinaceum, and whenever you have a gallinaceum in population, there's a mass mortality, actually. And birds don't get gallinaceum as well. But ducks, they do get some of these parasites, but not so much of Plasmodium and Hemoprodis, the leukocytosome. So again, it depends on the habitat they're using, what sort of vectors they have, and what they can get infected with, basically. Yeah. Yes.
So, do we have examples that a species got kind of extinct or like severely, the number goes
yes.
down? Because of
Hawaii.
extinction?
Hawaii had 95% extinction. There are very few endemic species left there. Yeah, that's the only example in the ever been. Yeah. I want some young women to ask questions as well. Come on, don't be shy.
The questions don't have to be science related.
My question is that regarding the recent Zika, you had shown some data. Do you have any idea about the dengue and chikungunya? Because both of these viruses are also transmitted by mosquito vector, that is the Aedes.
Aedes, yes.
So any idea about, not Indian data, but from some other country, do you have some data whether these viruses are also identified in birds, some other region, for example, in South America?
Okay, so that's a good question, actually. So no, there's a lot of hypotheses as well. People have done reviews, but no one actually has looked for these viruses in the birds, whether we call them like spillover infections or something. No, we don't have that information. There are some old literature, somebody has looked for antibodies and stuff, but to be honest, we have no idea. Nobody has actually looked into it. Yeah.
Hello, ma'am. I wanted to add on when you talked about Zika virus. So recently...
Oh, that's a mistake I did. I should not have shown that slide.
So recently, it's a news like microcephaly is due to Zika virus. So have you researched something about it on birds? I mean, is this problem also coming in birds or not?
So birds don't get Zika. I think you're misunderstanding. So the reason I showed Zika slide was about the way I am getting this fine-scale information on the parasite distribution as well as on their vector species. We need to do similar for other diseases as well in terms of disease ecology, which we don't have much data. Looking into, for example, Aedes aegypti abundance or population dynamics, so somehow we can do. Birds, when they are very highly infected with malaria, for example, we don't catch them. These are the ones which I catch. They actually have gone through the infection, so they have survived, basically. Yeah.
Thank you.
Yeah.
Hi, nice talk. I wanted to ask you, as you mentioned that the infection also improves the fitness of the bird, in a sense like those more infected are not been able to go back to their native place, wherever they go. So does it also evolve the species in terms of its fitness? Do you think that it also helped them to diversify, evolve also? Do you think so?
So I think so the migration has, I think, evolved around that, right? The birds which migrate in search of, because to avoid the cold temperature or looking for these resources, food resources, which are available in other habitats, because it's a part of evolution that has taken place. Now, in terms of the fitness cost, in my mind, the birds which are getting infected with a lot of parasites, their immune genes should be much more diversified than the ones which have never been exposed. So if they manage to live the infection and they go back, then they can transfer this gene. But here's one thing, the more infected you get, it slows you down. By the time you reach your territories to breed, there's a lot of competition. You may not be getting a good quality habitat to breed, and the competition is quite fierce. So yeah, so I think that's how it evades you out of the whole system, basically, from the population. Yeah. Yeah?
Yeah.
Thank you.
Thank you, Farah. Thank you so much.
Okay, so now all you young, the old ones we won't care so much about, but the young ones, close your eyes, imagine sampling birds. Does that inspire you? Does that inspire you to become an explorer? Come on, close your eyes. Oh, well, okay. So I'm your co-host with Sarah. My name is Ranjana, and we are here to give you a bit of a break while we transition, and so that you can switch gears. We are going to switch gears now, and the next up we have with us is Arun. Yep, Dr. Arun, SP Arun, and what you know already from the flyer is that he will talk to you about visual perception and object identification. See, there was a reason I asked you to close your eyes and imagine yourself already an explorer. But none of you are doing that. But what you don't know is that he loves optical illusions. And if you want to really look up, and that's another quirk, right? I mean, optical illusions are fun. And if you want to like find out more about optical illusions, do visit his lab website. It's very easy to find. Just Google SP Arun ISC and you will find him. The website is complete with scientific principles behind the illusions. The videos are hosted on his website. And you must take with you a bit of time and an explorer mentality to really, really enjoy them. He is also someone who is committed to discouraging single-use plastics. He's environmentally aware. And I would say he's driven to great heights. Welcome for us, all of us. Arun.
Thank you. Thank you to... Oh, I see.
So while he's figuring out his... Ask questions. I mean, they don't have to be particularly scientific. They can be like curiosity questions. Why do you do this? Why optical illusions? Why is your shirt pink? Maybe not that question. That's not a good idea. But yeah, I mean, like non-science questions. Ask.
Okay, so thank you to the organizers for inviting me. I look forward to all the questions. Please do keep the questions alive. So I'm not a biologist. I actually tried to avoid biology as soon as I could. So at 10th, you have this choice between maths, physics, chemistry, and so I avoided biology. And but I'm a neuroscientist now in a rather quirky department because we all come from very different backgrounds. There's no undergrad program in neuroscience in the country, so you tend to come to neuroscience if you're really interested. And I hope that this question is something that you'll find interesting. So. This is, I want to thank upfront our lab. We have an interesting mix of people coming from both engineering and biology backgrounds. Also, I want to thank all the funding agencies, in particular, of course, the IndiaAlliance, for all their support throughout my independent career as a scientist. Let me start here. I hope some of you have read books like these. I read them when I was growing up in school. And I was very inspired by all these sci-fi books about robots that live in society. There's also a movie by the same name, I, Robot. Some of you might have seen it. And they talk about a future where robots are actually coexisting with us and are programmed to have certain ethical rules. And Asimov is great in this regard. He talks about all these elaborations of these rules and beautiful universe, actually. But compared to where we stand today, and this is sort of how I got into neuroscience myself, contrast to where we stand today in terms of all these kinds of technologies, right? We type into machines instead of talking to them. We can't just talk to a machine and say, okay, set up a calendar event for me, or like, type out this email to my mom. Can't do all these things, right? Not yet. You tidy up your room instead of a robot doing it for you. Again, that's a big challenge for robots to just go about picking up objects without dropping them or without crushing them and navigate into a room and so on and so forth. But really the challenge that I want to talk to you about and what I really sort of started getting really fascinated by and continue to be fascinated is things like this, where we are still unlocking our laptops with a password instead of with our face. And that's partly because computers haven't really, or we haven't managed to make computers actually solve this problem of recognizing our face regardless of where we look, how we look, and so on. Okay, you might have seen these features in your cell phones and laptops, but they don't work robustly, and that's why you don't see them widely used today. Okay? And really the most striking example to me, and even now after studying this kind of problem for many, many years, is the fact that we still see these distorted letter recognition tasks when you browse a website and you're creating some sort of email account or doing something important on the internet. Usually you're faced and asked to recognize these letters. And these seem like a really, really simple task for us, but somehow they're actually there to prevent automatic computer programs from cracking this. So you can imagine that something that seems so simple for us to do, that is recognize a distorted letter, is actually a very hard task for a computer, and we don't quite know why. Okay, so I'll hope to tell you about this particular problem. Not sure what's going on. Okay, so why are computers failing so badly at a simple thing like seeing? Now, for those of you who are interested, there's some very interesting, there's a very interesting pattern that we've seen now with machines where it's called actually Moravec's paradox, where what this guy basically said, he was one of the early thinkers in the field of artificial intelligence. What he said was very profound, that the things that we find very difficult to do, like numerical calculation, setting up long programs, repetitive tasks and so on, are exactly what computers can do really easily. The things that we do very easily are exactly the things that computers find hard to do. Okay, so these are the simple acts of seeing, hearing, looking around, walking around. These are the things that actually are really hard for computers to do too. So hopefully I can explain to you why, a little bit of understand why this is the case. Okay, so imagine how you would actually go about setting up this kind of thing in a computer. And this is by and large how computers still do vision today. What basically happens is the task here is to find a person in the image, and these are actually slides from a colleague at Carnegie Mellon. He calls it the miserable life of a person detector. Basically what you do is, if you want to find people in the image, you do a very, very simple approach, which is that you have a template for how a person looks, or the average person looks, and you run that template at every point in the image. That's what is shown out here. These red windows are actually showing a window that is being scanned across the entire image, and wherever you get a good match, you say that's a person. Okay, that's how a computer, that's by and large still how computers are trying to find people in an image. Okay, so you look for a content on the window, you look for a good match, and if you get a good match, then you call it a person. So let's see how this kind of approach will, and you know, really what varies across computer vision algorithms is really the kind of match you look for and the kind of features you're looking for. Okay, but the basic idea is still the same. So then how does the detector do? Well, there's a couple of true detections, as they call, which is that there are actually people in the image and the computer has found it. And the reason it has found it is that the match between the template that the computer has with the template, with the content of the image is actually very good. So far so good. It finds all the right people in the image. But if you look at a little bit further, it also misses a couple of people. And if you look at the content of those windows, you will see that, yeah, and there's a couple of places where the person is not very obvious and the computer is missing it. And still, this is not looking too bad, right? Because obviously, these misses were actually blurry people, and sometimes you may not have found it and so on. So there's still reason to be happy. The place where it really starts breaking down is if you start looking at the false alarms, which is, for example, things like this, where the computer has basically discovered a person in this lamppost. And the reason why is because there's some kind of pattern that the computer has found which is very similar to a person. Well, now you can say, well, maybe I shouldn't look for a match like that. I should increase my threshold or requirement for a match, right? So that's very reasonable, and turns out that if you start increasing the threshold such that this is not detected, then you'll also miss a lot more people. So that is the trade-off here, which is that computers are not figuring out the right kinds of features that you need to pick out of the image to find a person. Whereas people, of course, are very good, and of course, no one in their right mind would actually find this to be a person. Okay? So when you look at the errors of a computer vision algorithm, you get weird errors which people would not normally make. Okay? So still, if you look at the performance of these computer vision algorithms, or even if you want to recognize the letter A or the distorted letter A and so on, you'll find that the performance of any given object detection task is actually almost 90 to 95%. So it's not bad. I mean, getting 90 to 95 on an exam is good, right? So the problem is that it doesn't scale up. So for example, consider what happens if you have a large number of categories you're looking for. If you want to know how many objects we recognize just routinely in the world, look for the number of common nouns. And if you look at the number of common nouns, there's at least about 10 to 30,000 common objects that we can all name. Okay, so if you look at that many different object detectors, basically what you're doing is you're looking for a chair in the image, a keyboard in the image, a car in the image, and so on. And basically, if you have a high confidence match, you'll say it's a correct detection. But look at what's happening now with this kind of image, where basically what happens is if each detector is having a 90% correct performance, okay, and you have 1,000 categories, we're not even talking about 10,000 or 30,000. With 1,000 categories, the probability of error becomes now actually 100 false alarms per image. Okay, so even though you have a pretty good performance for a particular object, scaling it up to a large number of objects actually starts breaking down. You will start having too many kinds of errors in the image. And that's really why computer vision is not really working too well today. Okay? So on the other hand, if you start looking at how the brain does it, and this is really the core focus of our lab, this is the monkey brain, and It's very similar to the human brain. It's used as a model system to study vision in the in humans. And the very interesting thing about the monkey brain, as well as our brain, is that a huge portion of your brain is dedicated to visual processing. Okay, consider how large it is. This is actually about 40 to 50% of your brain is involved in some kind of visual processing or the other. And if you think about the amount of space the retina occupies on your body, you can imagine how much more space is being occupied in your brain, purely because vision is so important and interesting, and it's a hard problem for the brain as well. So I want to show you a couple of interesting optical illusions which might give you an insight into how the visual system actually is doing all this complex processing. Okay, this is one which there's several that I had a choice of, and this is one that I like a lot. So if you look at the grayscale, if you look at the color of this object here and here, this one looks like a dark surface and this one looks like a bright surface, right? All of you agree? Okay, so now look at what happens when I obscure this corner. Is this looking dark and is this looking light? So the difference has gone away. They're exactly the same level of gray. Okay, so what's going on? When I turn it back, I've just shown you that the same image is actually the gray level here and the gray level here is exactly the same. Okay, but still, when I put this corner back to you, again, you should be seeing a dark surface on top and the white surface at the bottom. So what's going on? Why is your brain actually trying to fool you? Well, I would argue that the brain is not trying to fool you, because the job of your visual system is not to measure brightness in the image. Your job of your visual system is to figure out what is actually out there in the world. So if you look at what is out here in the world, you'll see that this part of the visual image is actually lit by a light falling on from above, okay, and it's not in the shadow, whereas this part is actually in the shadow. So if something in the shadow is actually as bright as something that is not in the shadow in the light, then the thing that's in the light must be actually darker, right? That's the inference that your visual system is actually doing to produce this kind of illusion. Okay, that's why, and this part of the image is actually a crucial part of that illusion, because it's giving you a clue about the lighting direction of the image. Okay, so what I want to really impress upon you is that your visual system is faced constantly with this problem of inference, because the content of the image is not strong enough to tell you what is actually out there, and your visual system has to constantly figure out what is actually out there. Okay, so that's illusion number one. The second one, I hope all of you have seen this. Please raise your hand if you have not seen this. Okay, okay, good. But you should have seen this by now. If you've been, you'd have seen this image, and I want to run this quick poll because I'm a vision scientist, and it's an experiment waiting to be done. So how many of you see this as white and gold? Okay, not a pretty big fraction. So all of you are my friends. Who of you who sees it as blue and black? Okay, so this illusion varies depending on the lighting conditions, okay? So, but the interesting thing about the illusion, and this is why it broke the internet, is that when you look at this illusion, when you see it as white and gold, you cannot believe that somebody can see it as blue and black, okay? The people who see it as blue and black will not believe that somebody else can see it as white and gold, okay? So what's going on? I mean, what's going on with our brain? What's the problem, okay? So here's an explanation that, here's one explanation, still an active area of research. If you have a question, please raise your hand, I'll be happy to answer it. But here's one explanation that vision scientists think that is actually responsible for what's going on. The idea here is that whenever you look at any image, you have to figure out the true color of an object. Now, how do you figure out the true color of an object? Well, you have to factor out the lighting color that is falling on the object itself. So for example, in a reddish kind of room like this, this is all reddish light. So the real color of the object might be obscured by the fact that light falling on it is a different color altogether. Okay? So the idea here that what's happening here is that this image is somewhat ambiguous. So, and the ambiguous image actually reveals your beliefs about the color of light that might fall on the object. So here's an interesting explanation from XKCD, very interesting comic, by the way. So here's the color of the dress. Now, inside your mind, if you have some implicit belief that light falling on an object is generally bluish in color, then blue light falling on a white and gold dress will actually produce this image. Okay? If you have some internal belief that light that falls on objects is generally yellowish in color, then yellow light falling on a blue and black dress will actually produce this image. So what your brain seems to be doing is to try and factor out the, or try and deduce the true color of the object while factoring out the light that's falling on the object itself. Okay, so if you saw it as blue and black, presumably your implicit belief is that light that's falling on, light that you've seen in your experience, or maybe it's genetically determined, whatever it is, you have some implicit belief that light that is falling is generally yellowish. Okay, and that's why you are able to see it as blue and black. Okay. Another example. Here's an image that we actually took a photograph of this in our lab, outside our lab rather. This is Pramod. He's not a midget. He's a real sized person, but he looks like a midget here. Okay, and even though I have this knowledge that Pramod is not a midget, I look at this and my visual system says, you know what? I don't care what you think about Pramod. He's a midget here. Okay, so what's going on here? Here's another view of this same display. We've just photographed the same scene from a different point of view, and this is really the main setup that we've shown here. Okay, so what we've done here is that the base of the chair is actually twice as big, but it's twice the distance from the camera, so that when you look at the right point of view, the chair is actually the base of the chair looks like it's sitting on the top of the seat of the chair sitting on the top of the legs. Okay, so then what's the brain trying to do here? Okay, so the interesting thing here is that the brain is basically saying, or rather the interesting thing here is the brain doesn't know the true size of an object. Okay, why is it that you don't know the true size of an object? Well, the reason is if something is closer to you, it produces a bigger image. So you fundamentally don't know how big an object is, because it could be just closer, or it could be actually a bigger thing but is further away. So how does your brain figure it out? Well, it's going to try and do all this calculation and inference to figure out how big an object is. And this is a case where the inference has gone bad because we've given it a very clever coincidence of cues. So what have we done here? We've said that at this point of view, the chair is looking like a plausible object. And if the chair is looking like a plausible object, then this object is actually right next to Saurabh, who's standing here. Okay, if the chair is actually a plausible object at this location, then the brain has figured out that this is the depth of at this depth, the Pramod is producing a small image. Okay, so if a small object is, if I have a small image on my retina, but the object is close by, it must be a small object. Okay, if it's a large image on my retina and the object is close by, then it's a large object. So you have to always figure out where the actual depth of the object is. So this is what your brain is doing here, and that's how it produces the inference that Pramod is a midget. Okay. This is even more extreme in this particular demo, and this really highlights the fact that your brain really doesn't know how big things are. Okay. Okay, so all of this is happening because your brain is trying to figure out exactly how far away something is and then how big something is. Okay, and all of these illusions, of course, are constructed very carefully by vision scientists to reveal these hidden assumptions that your brain is actually engaging in. Okay, it doesn't happen all the time because most of the time all these cues are in agreement with each other, and so it's very obvious how big something is. But sometimes, if you carefully construct these illusions, you can reveal what your brain is trying to do. Okay, so it may get kind of downhill and less interesting from here, but what I want to do in the time that I have left, how much time do I have left? Maybe, okay. So what I want to do now is to give you a sort of sample of the kind of things that we are doing in our lab. This is our lab at ISC Vision Lab, and you can read about it. We have some sort of infrequent Facebook presence, and these are all the things that we do in our lab. That is, we are trying to understand how vision works in humans. That's our broad general goal. I'm going to show you examples of how we use visual perception in humans to understand vision. We also do brain imaging in humans, so we take people to an MRI scanner. We can image the activity in different parts of the brain where they're looking at stuff. And very interesting patterns. We can compare the visual processing in different regions of the brain. We also record from neurons in the monkey visual cortex, And a uniting theme of all of this is that we're using computational models. So we actually use computer vision, compare with computer vision quite a lot, go back and forth and so on. Okay? So the question that I want to really put to you today, which we've been studying for a while, is to understand, we want to understand how objects can be, we want to see whether objects can be understood using their parts. Okay? So here's an example. We all see objects around us every day. We perceive the objects to be containing various distinct parts, and we simply wanted to understand, can we understand the percept of a whole object? And it's a very fundamental, sort of almost philosophical question. Can we understand the whole using the sum of the parts? And how do the parts combine to form the whole? Okay, so that's a sort of uniting theme that we've been pursuing across many, many different studies in the lab. And this is the not midget Pramod, who's actually doing the who's been doing some of these studies with me. So we started out with objects like this. The reason we started with objects like these is that we wanted to keep the two parts of an object very separate from each other. We thought that if you put the parts abutting each other, then you would create all these new junctions and features that potentially the perceptual system could be using. So we wanted to keep two parts of an object far away from each other. So that started out. We've of course done further experiments where we've joined them together and seen what happens. But this is a set that we started out with. And the idea here is that I can create a large number of objects using a very small number of parts. This is crucial because then we can see how the small number of parts might combine to create the whole object. Okay? So then we wanted to know, for example, we notice things of this sort. If you look at this pair of objects, these two objects look very similar to each other, whereas this pair of objects doesn't look so similar to each other. But logically, they're both equally similar because they differ in one part. They share one part and they're different in one part. So how do you go about actually looking at how the visual system compares objects or considers which object is similar to which? So to answer this kind of question, we've been using a principle that's actually found in nature. And again, I'll have a little visual experiment for all of you. Raise your hand if you find a leopard in the image. Okay? Might be a bit hard to find, but all of you guys are pretty good. This is the principle of camouflage. Of course, now there's no leopard to be found. But the key idea here is that actually the object is hard to find because it's similar to its surroundings. That's really the key principle here, which is that if you want to know, okay, so here's the leopard in case you haven't found it. It's very difficult to find because the markings on the leopard actually have evolved such that it's similar to its surroundings. Okay? And come and talk to me at the break or afterwards about, there's lots of interesting things to say about camouflage, including answers to several mysteries like why do many animals have white fur on their bellies. This is a reason having to do with visual perception. And this also interestingly tells us that all animals share many aspects of visual perception because leopards haven't really evolved to be hidden to human beings, they've evolved to be hidden to their prey. Okay? So, but what we've done in our lab, and this is a principle that everyone who studies visions often uses, is to abstract out this basic idea that if you take a long time to find an object, then it's very similar to its surroundings. So for example, in this display, the odd one out, there's only one odd one out, and there's identical items, and here the odd one out is a little bit easy to find. And that means that these two objects are very dissimilar to each other. That's the basic idea. But on the other hand, here the odd one out is a little bit harder to find, and that's because these two objects are similar to each other. Okay? So all of you must have experienced this as you look at this display. So then the interesting thing is, if I do many, many such experiments, I can construct a map of your perception, which is that I can tell you in your perception how similar you think these objects might be. So for example, this is a map that we've created. The idea behind this map is that two objects that are close by on this map are actually hard to find in visual search. This is exactly a pair of objects is an experiment that we've done. Okay? So you can see many interesting patterns, like all these objects that share the crescent actually are, how are we doing on time? Okay. So all the objects that share a part actually are close together, which means that if you have one of these objects against the other, then it's a hard search. You also see mirror images, that is that mirror images are actually close to each other. That's a well-known idea of mirror confusion. And the basic idea that we wanted to see in this experiment is to see whether we can explain all of this data using the fact that there's only a small number of parts that have created these objects. Okay? So this is the basic model that we've created to explain the data. The idea is that we have two objects, AB and CD. They're created from parts AB and parts CD. And we are just listing out all possible interactions between these parts. So for example, we thought that maybe the visual system compares the corresponding parts of these two objects. Maybe it also compares the parts at opposite locations in these two objects. And for completeness, we also thought that the visual system might care about the similarities of the parts within each object. That's the full model. And it turns out that this is a very solvable model. We can solve it using standard regression. And it produces a very good fit to the data. What I'm showing here is the observed data versus the predicted data. And the basic idea here is that the dissimilarity between objects can be explained using dissimilarities between the parts. Okay? And maybe I should kind of wrap up here. So I'll just summarize what we've been doing after that. So what do you do when you find a rule? Okay? So that's what I'm putting up here. What do you do when you find a rule that says that the dissimilarity between objects is explained using the dissimilarities between the parts? Well, you try to break it, of course, right? So we've been trying to break that rule now. So when you start breaking the rule, the most obvious thing we could think of to break the rule is to look at objects like these, which do not look like they contain parts. Okay? So you look at these objects and you feel like, okay, these are kind of symmetric objects. The two parts are actually, there's no boundary between the two parts. There's no separation between the two parts. So can we explain how these objects look in perception? And more interestingly, there's a long history in visual perception and actually even in art of the fact that symmetry is a very special property. So for example, we've been creating architecture of this kind. This is the Somnathpur temple in Karnataka. And we all appreciate symmetry in art. We appreciate symmetry in people's faces. We appreciate symmetry actually everywhere. So the question is symmetry something special in the sense that the brain realizes that the two parts of an object are identical and creates something that is more than the sum of the parts? Okay? And now we can actually answer this question because we can say whether symmetric objects are different from the sum of the parts or not. And to cut a slightly longer story short, we found that symmetric objects actually are the sum of the parts. So there's nothing special that's going on when the brain looks at a symmetric object. It is still behaving as the sum of the parts, but still that property of this kind of part summation explains how we perceive symmetry. So I think I'll kind of just wrap up there, skipping a bunch of things, but just to tell you that okay, so the brain uses a sophisticated code for objects. I hope I've made you think a little bit about vision. It operates according to simple linear rules. We found a couple of interesting rules. Trying to break that rule seems to lead to some insight, and that's kind of what we've been trying to do, establish how things interact and then try to see whether whether special conditions are there where they don't interact or they shouldn't be interacting in that way, and try to see if those are deviations. And then of course understanding this nature of this kind of sophisticated code might help us make computers see more like us. So thank you for your attention. I'll take questions. Yes.
So why can't we make the computer see?
Yeah.
What do you think?
So basically what I think is going on is that the visual system is doing, so in computers when you try to represent an image, you try to preserve all the detail that is present in the image. Whereas if you look at the way the visual system works in our brain, it's actually not trying to preserve all the details. It's actually trying to throw away certain details in the image so as to make feature processing and object recognition more efficient. And we haven't quite discovered what the format of that representation should be. So if we do figure it out, maybe a computer vision person might figure out what's a good way to represent the image. Maybe a vision scientist or a person who studies, a neuroscientist might figure it out, but I think that's up for grabs. That is that we don't really know what is the format of the code in which the brain is using to represent objects.
You also talked about symmetry.
That's right.
So symmetry is quite prevalent in nature.
Yeah.
So what do you think? What might be the reason?
So what we have found is actually that symmetric objects tend to stand out. Compared to asymmetric objects, symmetric objects actually tend to become a little bit more distinctive in perception, and that's a measurable effect. And that's really what we think is why symmetry is becoming more special. And what we've actually shown is that the time you take to detect symmetry in an object can be related to how much it stands out compared to its surroundings. And that we think explains how very, very regular objects like circles and so on are considered highly symmetric because they have many self-similar pieces in them. And that's why I think, at least that's our provisional explanation for why it's that case.
So my question is, according to you, if computers are not so much perfect in doing things, even after so much advancement in computer technology, can we expect in future that computers will dominate over humans in future and will replace humans at the place of work? Can we rely on computers?
So I think we are very, very far from such a future, although there are lots of people who have imagined such a future, but we're nowhere close to any of that. What is crucial here is, I mean, in the early days of AI, there were a lot of people who thought very deeply about how computers might change the way or how they may emulate human thinking. And we don't understand how many of these things happen in the brain. And for instance, the early thought was all of the question of artificial intelligence boils down to this question. This is a quote by Douglas Hofstadter, who said, what is the letter A? That is, how do we understand when something, if you look at a distorted version of the letter A, how do you recognize it as the original letter? Well, we're very far from the future where computers might dominate. But they are already dominating us in terms of calculations and various kinds of numerical processes already. So it's not like they're not dominating in certain other fields.
Okay, thank you.
In the interest of time, we'll take one last question, sorry, and we will let one of the young generation take it.
Sir, you just said what is the letter A? What is the letter A? So how do we perceive different handwritings? Yeah. Like in A, I have written something and you have written something. How do I perceive that they are the same thing?
So it appears that the brain's able to generalize across distortions of some particular shape. And when you look at these captchas, those distorted letters on websites, you don't see the same distortion ever again. And you might not have seen that particular distorted letter ever before also. But the reason that you're able to recognize the letter A is because perhaps you're looking at some diagnostic sort of features in that letter. We don't know what those features are. And so we can't really say what is it that actually, but there's apparently a family of distortions that we can handle even in faces. Like you can take a face and actually even just take open it up in some image program and squish it heavily. You'll still be able to recognize your friend, and you'll be able to do quite a bit of recognition even though they're very distorted. So the family of those distortions that we can handle is not known. Okay.
All right, thank you so much, great. Arun, for that fantastic and very, very interesting talk. Arun is going to be around, so I saw a lot of hands up that side, so please feel free to grab Arun at lunch or at the break outside. Right, so next up we have someone who runs marathons, is also an artist. You'll be able to see her painting in Jacaranda room, it's on display, and also a scientist. So we have, our next speaker is Dr. Rashmi Rodriguez. Rashmi will talk about her journey from being just a beneficiary of mobile phone technology to harnessing it to support healthcare. Rashmi is interested in developing better public healthcare support systems and works on infectious diseases like HIV infection and tuberculosis. So over to you, Rashmi.
very good morning to all of you. Thank you for coming over to listen to all of us. Okay, so I have come all the way from Bangalore and prepared a fairly basic presentation for all of you to listen to, and I hope at the end of this, I will at least be able to communicate a simple message to the young crowd, right? Okay, so how many of you have mobile phones? Everybody, I'm sure, or at least you know how to use one, yeah? Okay, so if you think about your phone and what you do with it, I'm sure at some point in time, you have used it for some healthcare-related activity. You've at least Googled a disease, perhaps, right? Spoken to maybe your doctor, correct? Yes. Or maybe used it for physical activity when you tried to look at the number of steps you walked for the day, right? So some of us may have done even that. So I'm just going to take you through my work with mobile phones and how I have tried to harness them for, or to, kind of to improve healthcare, right? So here is a picture that shows the evolution of the mobile phone, just like the human evolution. Now, just as my previous speaker mentioned that generally objects evolve to match their surroundings, so has the mobile phone. It was kind of not very pretty in the beginning, and now you see it's quite handsome or pretty, right? Just like the humans today. So, I would not be wrong if I actually said humans nowadays are born with a mobile phone. Right? Well, in some way or the other, the conceived fetus, right, interacts with the mobile phone at some level. And so, the young children that are born nowadays are quite intuitive when it comes to using a mobile phone. At the age of 3, you would have seen children playing kind of games on the mobile phone, yes? Right. So, well, it's now in the genes. Perhaps the genetic code has changed to incorporate the mobile phone. So here's a picture. The top part of it shows the Delhi telephone exchange in the 1950s. A lot of humans there. Now, it's kind of, okay, so now it's been replaced by an entire web system of servers and networks. So you don't really need a human component. If you look at the growth of mobile phones itself, right, both globally and in India, the numbers of mobile phone users have increased tremendously. I remember getting my mobile phone somewhere in 2004. I had gone home, opened my dad's cupboard, and there was a nice little box with an Alcatel mobile phone. It was given to him by somebody. And I wanted it because generally nobody else at that time actually owned one. And he was kind enough to give it to me. And I literally went around showing it off to people saying, oh, I have a mobile phone. And at that time, that mobile phone, unlike the other mobile phones that people used, had a speaker in it. So it kind of boosted my morale in a way. But what I want to bring home with this slide is, if you look at the picture in the background, that is something that people laugh at, especially at IT conferences or mHealth, mobile health conferences. They say India is a country that has more mobile phones than it has toilets. People may not use a toilet, especially in rural India, but they will surely have a mobile phone. The point is this: mobile phones are so common that you can actually try to deliver healthcare in some basic way, even to people in the most remote villages today, right? And this is an excellent tool that we can harness in a country that has so many people and so little healthcare resource. So this was one of my early works in mobile healthcare technology. We did a survey in a village. And what I wanted to do was, I was really happy that I was able to successfully do the survey in 2 months. I wanted to present it at a community medicine conference. So the student who worked with me on this sent this paper to the conference as an abstract. And the organizers laughed at it. They said, what is this? Why are you kind of talking about whether you're making calls to a doctor, whether you're not, whether you're sending an SMS, whether you're not? I mean, what kind of a paper is this? It actually didn't get accepted as an oral presentation, but we just had to convince the organizers to accept it for a poster. And of course, it got accepted at that time for a poster. I was, of course, able to publish this paper later in a nice journal, and it was well appreciated. But yes, it was part of my work, and that's somewhere where I began. If you now look at the different things mobile phones can do, Apple has come up with technology to kind of monitor your heart rate. You can speak to your doctor. You can see your doctor on the mobile phone if you want to, maybe if you have a suitable app. Mobile microscope technology is also available. And there are people who are now trying to use it to diagnose malaria in rural India, not rural India, but rural parts of the world. So you can have ultrasound that you can view on a mobile phone. And of course, you can have you can order your pills and medications on a mobile phone also. So then, when I started my work with mobile phones, the disease that was considered of immense public health importance was HIV. There were a lot of people suffering from HIV at that time, and nobody really knew how we could help them. What was important was that they took their medications adequately. They had to take at least 95% of their medications if their disease was to be under control. Keeping their disease in control also prevented them from spreading the disease to other people. But the question was, would people with HIV take their medications correctly? And can we help them make sure that they took 95% of their prescribed medications correctly, right? Now, by itself, people with HIV are largely driven by the motivation to take their medicines, because they know that if they do not take their medications, they will not live very long, right? So, of course, they want to do it. And so, when we kind of approached patients with HIV, asking them if they would like to have a mobile phone application to help them take their medications better, they really loved it. So we developed kind of a basic mobile phone application. This was somewhere in the year 2010. Patients with HIV still had very basic mobile phones, right? And when we spoke to them, we realized that they did not like SMSs so much. What they liked was voice calls. So unlike what people had done in Africa, where they had used SMSs to remind patients to take their medications better, in India, we had to think differently. And we came up with an interactive voice response call. This went out to the patient once a week, right? And it kind of told patients to press 1 if they took their medications, and 2 if they did not take their medications. Now, just to add another component of the reminder, we sent them a pictorial SMS. We had to take care of the sentiments of the people with HIV. And that is because people do not like others knowing that they have HIV infection, right? They may be stigmatized. They may be ostracized, for example. And therefore, they were very concerned. So we had to take away all mention of HIV in whatever we did. So every week, patients received an interactive voice response call, and they also received a pictorial SMS at on two different days, right? And what we did was we put this in something called a clinical trial. Do you know what a clinical trial is? Yes? It's kind of an experiment that you would maybe conduct in your physics or chemistry lab, except that it's on humans, right? So we put this in a clinical trial where there were one group of patients that did not receive either the SMS or the IVR call, and another group that received both, right? And in the end, we wanted to see whether this concept of giving patients a reminder was ever useful. So, unfortunately, right, this study went on for about 2 years. Unfortunately, our intervention was too simple. It's like you get several SMSs on your phone, or you get several IVRs. Initially, you're extremely enthusiastic about them. You answer them, especially if you had a new phone, right? And after some time, you kind of don't like them so much anymore. You're like, why are these people calling me? Why am I being pestered all the time about maybe my medications or something else? Why am I being reminded so much? So overall, in the end, we realized that the intervention that we had developed it didn't really help patients very much. Also, another reason may have been that it did not have a human component, right? It was entirely a system, a web-based computerized system that made the calls. Unlike somebody calling and speaking to another person in person. So there may have been a difference there. A third reason may have been because HIV patients were motivated enough to take their medications by themselves. They were really worried about the consequences of their disease, and so they really took their medications really well. So, but the question was, we have a tool that we can effectively use. And how could we actually use our experiences, maybe, to make things better, perhaps for another disease? Is there another disease that could benefit from the experiences we had with patients with HIV? And then, of course, there was tuberculosis. It is another disease that also affects a large number of people, especially in India. It also requires patients to take medications for maybe 6 months. And as patients are not as scared about the consequences of the disease, they tend to falter a lot with tuberculosis medications. Now, what system we currently have is to observe patients when they take their TB medications, right? You have to say that a patient has taken their medications. And for that, the patient has to come to the doctor or to a person designated to observe the patient while they take their medications. And that is called direct observed treatment. Okay? So now with mobile phones and our experience, the question was, can we do something else? So while I was working with HIV, there was a group, Microsoft Research India, and 2 researchers there, Bill and Andrew, who approached me with a concept that they had. They said, we want to do something with TB patients. We want to see if we can help them take their medications better, but we don't really know how to go about it. And so we sat and discussed around a table about giving a patient a missed call, right? And then that evolved from a missed call. So initially we thought maybe we could, give a missed call reminder to the patient, but what we wanted to do was actually to record whether the patient had taken a dose. So what we did is, we sent the patient an SMS with a number on it, with half the number on it, and kind of with the number on it. to which the patient gave a missed call to say they had taken their medications. It was not necessary for anybody to answer the call. That was when the intervention was very basic, right? So a patient would receive a mobile phone number via a message and then may give a missed call to the number and hang up. So we would record that the patient had taken their medication. Now the concept has evolved further, wherein there are nice, neat little packets available to the patient, and the patient flips the cover on each of the medications, and there's a number that's relieved. So that's revealed. So what happens is every day the patient is expected to take maybe either 2 or 3 medications depending on their weight. And when that happens, there is a toll-free number that's there on the back of that slip. And the patient gives a missed call to that number, which is recorded. And then you kind of put all the missed calls together to see if the patients have taken all their medications, their prescribed medications or no. This is used with patients who have HIV and TB co-infections. It is not yet used for just tuberculosis patients. The reason being, we have a huge burden of the disease, and perhaps our healthcare system is not yet well equipped to take on the responsibility of removing completely the human component and giving it to a missed call system, right? But we don't know yet where this will go. Perhaps it will be used for patients with TB in the country, perhaps not. We're yet to see that. From there, I came across a group of researchers at Johns Hopkins University that were working with the concept of video DOT. Now, video DOT tries to look at the patient while they are swallowing their medications. And for that, there's an app. So what the app does is, what the patient does is, uses the app and records themselves taking their medications. The app transmits that video to the healthcare provider to view at a time when the healthcare provider is free. And they check whether the patient has taken all their medications. So the issue that we had initially when we discussed with Johns Hopkins University was that the app that was developed in the US didn't really work in India. Our mobile phones were perhaps of a different kind. I myself do not understand why, in a way. So we had to think differently, and we had to develop our own mobile application. This application is now available on the Google Play Store as v.cure. So, of course, this is available free of cost for download for patients to use, but they have to be registered by their healthcare provider on this application. Here's kind of a brief video that you can view.
With any health problems, completion of the full course of treatment is very important to ensure complete recovery. Through this mobile application, we aim at supporting you through your long months of treatment. Let me run you through with how this application works. Your first step would be to find a bright room with good lighting so that your video is clear. Once done, please place your phone in an appropriate position to record your video. And then click on the application. Language options will appear as the first screen. Click on the language of your choice. Then comes the login page. Here enter your user ID in the user ID box and your password in the password box. Following this, you will be directed to the instructional videos page. By clicking on the play button, you may choose to watch the video, or else you may skip it by clicking on the skip button. You will now appear on the recording page of the application. Here on the bottom of the screen, you will find a record button. By clicking on this, you may begin recording your video. Kindly show the tablets to the camera before you take them. Now you may take your tablets one at a time or all together, as I am doing in this video. Once done, please show your empty mouth to the camera. By clicking on the stop button on the bottom of the screen, your video will automatically be uploaded. Let me run you through with a few more features of this application. For instance, by clicking on the I button, you may get an idea about the number of videos uploaded and accepted. By clicking on the back button on the top left corner of the screen, you will be able to watch the instructional video once again. By clicking on the logout button on the top right of the screen, you may log out of the application. I hope that this video was helpful. Please remember that it is very important to complete the entire course of treatment, failing which the symptoms will recur with more severe complications and a longer duration of treatment. We hope for your successful treatment and complete recovery. Thank you.
Yeah, so this is the concept of the app that we have developed. And basically what it does is helps the doctor view the patient taking their medications. Yeah, you may ask, but the patient might spit out their medications, or they might kind of use a glass of fluid that is not clear, for example, milk, right? They could, and they could spit their medications into that glass of milk. Or they might use a glass which is like a steel glass, and they might still spit out their medications into it. Of course, these are the issues that we have. And I do agree that we may not be able to counteract all of them. The issue, of course, is that we can suggest to patients to use a clear glass of fluid and a clear glass itself to take the medications. But then, perhaps we're going to be a little too strict with the patient, and they may not like it. So we have to actually kind of come to a balance between how much we want to do, right? Because ultimately, it's the question of the confidence that we gain from the patient. We need to believe the patient to a large extent if we have to help them take their medications better. None of us like to be monitored closely. We kind of, step back a little bit. If our parents kind of tell us, where have you been till 11 in the night? We don't like it. So it's the same thing with patients that we have. So what we need to do, basically, is to try to be nicer to our patients, and that is what we're trying to hope to do with the app. Along with that, we're also trying to support them with a certain amount of counseling, which we didn't do when we did the HIV study, right? So we try to speak to the patients about taking their medicines better, what their problems are, and how they could kind of overcome those problems. We are, like the HIV study, trying to do another trial with this application. It may be successful, it may be not, but it will surely tell us, give us useful information on whether this can be used large scale or not, right? So that is the purpose. Is the intervention that we develop useful enough for maybe 1 billion people, right? Will they accept it? What about the intervention makes it nicer for them to accept it? And that is something that we constantly have to keep thinking of as we work with patients, with healthcare, and with the mobile phone. And nothing stops anybody from coming up with a new idea or a concept. So maybe as you all go away from here and look at your mobile phones, perhaps you will come up with another idea which may be one of the best, which has never been tried before, right? So as you go away, keep thinking to see where you can go, and that's important. So yes, I like to use this picture in most of my presentations, and it kind of gives me a sense of bringing the country together, and perhaps the mobile phone can do it, perhaps it may not, but yeah, let's hope for the best. And of course, I thank IndiaAlliance and my institution and other people who have helped me with where I am now. So, any questions?
In the interest of time, we'll take 2 questions. Sorry about that, but they're available so they can take it.
Hello. My question is, mobile phones have adverse effects, that is effects of radiations on human health, but mobile phones has also become a necessity for our daily lives. We cannot think of our life without mobile phones nowadays. My question is, what are the various ways by which we can continue using mobile phones as well as preventing adverse effects on human health?
Yeah, mobile phones are a double-edged sword, right? So you have benefits as well as ill effects. For example, alcohol also has benefits and ill effects. If you take too much of alcohol, you're sure to have a problem one day. It could be cirrhosis or it could be something else. So similarly with mobile phones, it is left, I think what is important is that each individual learns to control his or her use of the mobile phone, right? So you need to have restriction, you need some restriction about what you do. I mean, a good, what shall I say, too much of a good thing is bad. So I think it all lies with the individual.
You know, as you have informed us about the HIV, the problems like, you know, diabetes and sugar, how this can be used on large scale, because India is the capital of diabetes. And my second question is that when we have MTS, medical transcription system, on computers, in America it is used on large scale, how it can be used in India on the base of computers to benefit the patients? Thank you.
Yeah, so when you talk about diabetes and hypertension and non-communicable disease, there are trials going on in India that are trying to look at how you can improve physical activity and prevent, what shall I say, the onset of diabetes in a way. So there have been studies that have already come out that giving patients, educating patients via their mobile phones are helpful, and it can delay the onset of diabetes, while there are other studies that are currently ongoing to maybe look at different aspects of what can be done via mobile phones. I think Arogya World is one such group that is kind of trying to look at the use of, I think, health education via mobile phones in the IT sector, right, to kind of delay the onset of diabetes and non-communicable disease. Well, coming to medical transcription, I'm not really sure at the moment if we have, we probably have a number of people who have been doing medical transcription. Of course, in St. John's itself, where I come from, there was a unit that was trying to do it at a distance. So, yeah, I mean, I haven't really ventured into that field, so I'm probably not the best person to talk about medical transcription. But what is currently being done is they're trying to link or create a database or create a linkage between records and develop something called a health information management system for the country, which can, in a way, perhaps, if you are ill now, and you want to go to another place, or you have moved to another place, you've had surgery in one place, say in Bangalore, and for some reason you move to Delhi one day, and you need to go back to your doctor, and the doctor needs to know what your issues were previously in order to treat you now. And that can be possible if we have in the country a very good health information management system and something called electronic health records. So if we can kind of develop that and incorporate electronic medical records into the entire system, I think it would be a good idea. I mean, people are working towards it. There are groups that have come up with different solutions. We are yet to know whether that's going to be, I mean, yet to try it in the field, I think.
All right, let's thank Rashmi. Give it up for her. Come on. So are you having fun? It's not a usual question we ask in a science conference, but this is not a science conference. This is Explorer series, and these are our rock stars. So I can do my best imitation of a rock star thing. Are we having fun yet? Up there, the balcony people, are you having fun? All right, let's show how much we're having fun. And welcome our next and the last of the rock stars, Dr. Abhi Vanik. He is in a typical style of exploration. His story has many shades. His academic journey, in his own words, not mine, has been one disastrous stumble after another. Him starting with a failed project on bats, which then led him to work on tigers for his master's project. For his PhD, he wanted to work on tigers, but had to end up having to work on lions. Like, I mean, is this a stumble after a stumble? Like, this is how they explain stumbles. Like imagine, like, what a wonderful journey that is. See, I'm telling you, like, these are the way we are going to inspire you to, like, work with tigers and lions, or maybe hens and whatnot. So when he found that, the story doesn't end there. He found that even that project was doomed. That's a very typical thing that happens in science. So he gave up on the cat family, and he decided to find something non-charismatic that he calls and completely apolitical, and he switched to studying foxes. You know the cute little things that look really, really pretty jumping into the snow? No, not those foxes. He'll tell you about the foxes. So when he was actually working with foxes, Abhi met the domestic dogs. Yay. And then he found that they have widespread menace and spread rabies. So he will tell you, like, he'll pretend like he doesn't like them, but that's, again, not true, because he does have his love for the canine family and one particular member that he calls Permit. And that Permit has been his lucky charm since his first Permit. And he also has a long list of experiences. He's monitored olive ridley sea turtle nesting in the state of Odisha. See, life can be very interesting if you get into science. You don't have to be sticking to the bench all the time. You can go look at turtles. And he is also skilled in catching crabs, wild crabs, not farmed crabs. Let's give it up for Abhi. Yep.
Great. Ranjana has given my talk, so I'm done. Great. Thanks, guys. See you. Well, thank you, Ranjana, and thank you to the IndiaAlliance for inviting me to be part of this wonderful Explorer series on that decade of discovery. So, yes, let's start with some tigers, right? And it all goes downhill from here. After one more slide. You'll notice that one of these tigers is wearing a nice little necklace around her neck. That's a radio collar. These are not the tigers I studied. I took this picture about 15 years after the ones I studied because I didn't have a very good camera then. But my tiger study after my master's went downhill. So then I switched to lions. These are Asiatic lions in Gir. And as you would have read in the news recently, there's a CDV epidemic going on, or there's some epidemic going on. People are claiming it's CDV. Canine distemper virus that's threatening this population. Unfortunately, I couldn't study them. I did study lions later on in Africa, but I moved to these little fellows. Okay, these are Indian foxes, beautiful. They're quite charismatic, I think. This is a photo taken with a camera trap. These are little remote cameras you leave out in the field. And they take pictures by themselves because they're motion activated. Wildlife scientists use that very often now. In fact, these are sort of animal selfies, if you will. Well, while I was doing all of this, I started a PhD at the University of Missouri, and I couldn't escape tigers there either. The mascot of the University of Missouri is Truman the tiger. So I ended up having to talk to a bunch of people about how I didn't study tigers. Anyway, I studied foxes. And what we did is we caught them and we put radio collars on them, these little devices that help you track these animals. And as we were tracking these in the field and trying to find out where they were, we realized that the most common animal that we were seeing in the field were actually domestic dogs. These were everywhere. Now, domestic dogs, humans have a really long relationship with them. This if you will see, is a fossil. This is from the National Museum of Israel in Jerusalem. This fossil is about 14,000 years old, okay? But this lady here is cradling a dog. She was buried along with it. And this is the oldest evidence we have of the domestication of dogs. So this is the oldest fossil evidence we have. We domesticated dogs probably about 30,000 years ago. Or some hypotheses suggest that they domesticated themselves, that wolves sort of started hanging out with humans for scraps of food and so on. And the tamer ones eventually were domesticated as dogs. Okay. Currently, dogs are everywhere that humans are. They have a global distribution. India has about 60 million odd dogs. Okay? And wherever dogs occur currently, they're human subsidized. What does that mean? It means they depend on us for their survival. That if we were not there, then dogs would not be able to survive in the wild. There are very few true feral populations of dogs. That means dogs that are living without human assistance or intervention. Okay? The global population of dogs. So this human subsidy has really been good for dogs. There are close to 1 billion worldwide, okay, which makes them the most common carnivore in the world. Okay? So dogs are still wolves technically. They belong to the same species, Canis lupus familiaris. So there's about 1 billion of these animals all over the world. And across the world, they have a really good relationship with humans because they are our best friends. Okay? And including in India, in most places, we have a good relationship with them. So while I was doing my PhD study, we decided to understand the role of these animals in the interaction with the rest of the animals in the environment. Okay? So we started conducting vaccination camps in villages. So here we're giving a rabies vaccination to an own dog. We put a collar around its neck. I also fit some radio collars on these animals to see where they were moving. Okay. And then we started testing them for rabies, because rabies, as most of you probably know, I mean, who does not know about rabies here? Is there anybody who doesn't know what rabies is? I think everybody knows what rabies is. Okay, so we started testing them for rabies. Okay. And then we had one dog that was brought by a group of children much like this, okay? And it was completely asymptomatic, but it ended up testing positive for rabies. So this got me thinking eventually about this very deadly disease, because remember, rabies is completely fatal, okay? The origin of rabies and the knowledge of rabies is very old. The group of viruses that rabies comes from is called the Lyssa virus family. And in the Greek, Lyssa, the word is from the word violent. Okay, so it was associated with violence even by the Greeks. In Latin, rabies, rabias, or rabies, or rabio comes from rage, madness, fury. Okay, so all of these are words that were associated with this disease. There is some thought that actually the word came from Sanskrit, rabhasa, to do violence. Okay? And in fact, probably the oldest definition or the oldest mention of rabies comes from Maharishi Shushruta, okay? From the Shushruta Samhita, which is a Vedic text written probably between 1750 to 500 BCE, okay? So if you think about what a rabid dog looks like, or if you Google rabid dog, this is the photo that you will most likely find, okay? You might also find this photo because this is a very famous one from a textbook, okay? Or you'll find photos like these of dogs covering in the corner. Okay? That one is actually not a rabid dog. This one it's just an angry dog or just snarling. But that's the photo you'll see most often. Okay. But what does a rabid dog look like? So here's one video. That one is turned you can see it's very disoriented, okay? not sure what it wants to do, right? Walking around sort of on this, because rabies is after all a new disease of the nerves and the brain. Okay, it affects the brain, it changes behavior, okay? It causes discoordination. Eventually it causes a failure, multiple organ failure, okay? And this is what the offending organism looks like. This is the rabies virus. It's a bullet-shaped virus, okay? And in the words of Bill Vasic and Monica Murphy, it's called the world's most diabolical virus, okay? And our history of our association with rabies has really left an indelible impact on us, okay? But there's good news. The World Health Organization and many other organizations across the world, such as the Global Alliance for Rabies Control, have started a campaign that says, you know, 0 rabies by 2030 across the world. At the moment, about 60,000 people in the world annually die from rabies. Okay? Of those, most of these deaths occur in developing countries of Africa and Asia. Okay? India has the highest burden of rabies globally. We account for 1/3 of all of those human fatalities, about 20,000. Okay. The WHO then assures us that eliminating rabies is actually not that difficult, and I agree with them. It isn't all that difficult. Okay, but so there are, say, 5 easy steps to control canine rabies. It all depends on what's called mass vaccination, because we have very effective vaccines to control rabies, okay, both for humans and for animals. The main vector of rabies, or the main host, are dogs. Okay, so if you can control rabies in dogs, you can control rabies or you can prevent rabies in humans. Even if humans do get exposed to rabies, you can still have effective post-exposure treatment or vaccination that will then prevent you from getting this disease, this dreaded disease. Because remember, if you get rabies, there's no treatment done. Okay, it's 100% or nearly 100% fatal. There are barely a handful of survivors, and even those who've survived after very, very long treatment have been left with some handicap or the other. Okay, so what are these steps? One is to estimate dog populations. Okay. Then to conduct mass vaccination. Why do you need to estimate the dog population? Because you need to cover about 70% of that population to vaccinate them, okay? And if you cover 70% of the population annually, then you will achieve between 25 to 40% of what's called herd immunity. Herd immunity basically means that if you cover enough portion of the population, then the chain of virus circulation within the population will stop. Okay, so there won't be enough uninfected or susceptible animals for the virus to affect. Okay, then you assess the population coverage, and then you test the efficacy of your coverage. Okay, so as I said, India has the highest human rabies cases in the world. Okay, about 20,000 deaths, human deaths per year. Okay, there's an estimated. There's an estimated 20 million dog bite cases every year. So basically, somebody is getting bitten by a dog every 2 seconds in this country. And somebody is dying of rabies every half an hour. Okay? When you compare to other cases, you know, other causes of mortality, it's actually not that high. It's less than 0.01% or 2%. Okay? But it's still an important disease from the public health perspective. So let's look at this puzzle of rabies, okay? So you have your main reservoir host, the domestic dog, and for any public healthcare practitioner, that photo is a nightmare because that's the dog eating a fruit bat, okay? Fruit bats, I don't know if you guys kept up with the news recently. There was a Nipah virus outbreak in Kerala. Fruit bats are the main reservoir for that. Anyway, you try and understand population size, demography, the ecology of the species in general, okay? And then you understand the epidemiology, what's going on with the disease within the population, okay? You do that by doing surveillance. And then you have prevention and control, okay, of human rabies, of canine rabies, and then you test the effectiveness. And eventually all of this will feed into some policy that will help achieve this goal of 0 by 2030. Okay? So how do we go about doing all of this? So the study that the India Alliance is very generously funded, that we're doing now, is trying to find out and try and piece together or get more information about some of the pieces of this puzzle. Okay? In India, most dogs are free roaming. Okay? So we don't have all our healthy dogs on leashes being walked and taken to the, there are some proportion of that, but most dogs are free roaming. They live lives on the streets. This photo is from Bombay. Okay? But not all the dogs are the same, right? You have dogs in cities, as you can see around here in Delhi or any big city across India. They live mostly on the streets and are dependent on garbage or on handouts by people. You have dogs in villages which are owned by people but are still free roaming. So they're farm dogs or they're village dogs. People will give them pet names like Tommy or Bholu or whatever it is, right? And take care of them, but they don't give them vaccinations because that's not part of the, the education or the ethos of those, because those animals really don't have value. There's plenty of animals to have around. Then you could have the village free-ranging dogs. Again, very similar to your city free-range dogs. And then you have your farm dogs. Okay, all of these different categories of dogs. But they're all not the same. So what we did is we put a bunch of GPS collars on dogs around villages. So this is an area in Baramati near Pune. And you can see that some dogs move a lot more and some are very stationary. They're like right there in the village. They don't move. But some are sort of moving all over the place, covering much larger distances. Okay? So this tells us that these dogs behave differently, that these different categories of dogs have inherently different properties about them, so they can't be treated as one similar kind of animal. They're all different. Okay? There are other animals also out there. Dogs are not the only reservoirs of rabies. You can have species like jackals and foxes. Okay? So what we did is we also caught a bunch of these other species, for example, jackals, and we fitted them with GPS collars and we tracked them. So this is four foxes in their home ranges doing their thing. These are a few jackals. Okay? And canines are not the only. Remember, any mammal can get rabies. Even birds can get rabies. There's a known record of a chicken that got rabies. Okay, so imagine being pecked to death by a chicken. But chickens can't transmit rabies, thankfully. So we have the third most common carnivore in that landscape, the wild carnivore, the jungle cat. It's a small cat. And then so we caught a bunch of jungle cats and we put GPS collars on them. And then we found one which is living very close to a city and it's actually going inside the city at night. Okay? So there's an urban jungle cat walking around out there. And all of these animals are also part of this broader ecosystem. Because remember, there's lots and lots of dogs everywhere you look. In these villages, there are dogs at about 140 dogs per square kilometer. Okay? That's a lot of dogs. So we put them together in this map, and you'll see that this is in red is the jackal, and let me play that again. In red is the jackal, okay, and the dogs are in blue, and you can see that there's overlap in their movements because they're all occupying the same habitat. Okay, so that means there's a probability of transmission of rabies from one species to the other. Okay, and these are the kinds of information that we're trying to figure out now. Okay. So now we put all of those home ranges of those animals together, and remember that these are not all the dogs that we've collared. There's lots more dogs out there that we don't have information on. Okay, and we've collared a lot of dogs, about 40, 50 dogs right now. Okay, and about 30 wild animals, but there's still a lot of holes. But you can see that there's a lot of overlap. Okay, here's a 3D representation of the same thing. So you can see that these animals are occupying more or less the same space, and there's all high potential for interaction between them. So it sort of makes makes the, understanding the ecosystem of rabies a little bit more challenging, but also very interesting, and therefore allows us to find out if there are specific pathways that we can target, okay? So we started doing some surveillance to see where rabies is. I work with this organization in Pune called the Rescue Charitable Trust. They're an excellent organization doing great work. So we looked at the city of Pune itself and said, let's see how many rabies cases we find there. So we did something called passive surveillance, okay, where if somebody reported a potentially rabid dog, they would go and pick it up from the streets, bring it to their rescue center, and test it for rabies, okay? The opposite of that is active surveillance, where you go out and just test dogs, whether they are symptomatic or not. You just test general population at random, okay? So we did this passive surveillance, where we would go out and catch dogs that somebody reported. We'd then take a blood sample from them, also take a salivary swab from them, okay? And then in the field, we would this is their rescue center in Pune, where all sorts of dogs are brought in, and they're kept in this isolation unit. Okay. This dog turned out the previous one turned out not to have been rabid. This unfortunate puppy was bitten by a rabid dog, so it was very badly injured and had to be put down. Okay. Then we looked at the incidence of rabies cases. Now, you'll see that every month, okay, we've started recording them. And very soon, and this is the city of Pune, yeah? Over the last year, these are the number of rabies cases we've recorded, and it seems to be spreading out from the city. So 318 suspected cases were tested. Over 167 canine rabies cases were detected. Okay, we even had 3 bovine cases because rabies can transmit to other mammals. So 3 bulls were recorded. Rabies also manifests in 2 different ways, or there's actually several. There's a continuum between furious rabies cases where you have this classic animal going around biting lots of people, and then you have the dumb form where they just sit in one corner and die. How much more time do you have? 32 people were actually even bitten by these animals, but thankfully there were 0 human cases thus far, and that's mainly because Pune city has, it's a large city. It has enough vaccine available. The doctors know how to treat a victim of dog bite. So then the problem of human rabies is kind of controlled in Pune city, even though there's lots of rabid dogs. But that's not the same for the rural areas where most of our rabies cases come from, okay? So what we're trying to then understand is if we can find out this model of transmission, okay? Imagine there's an urban area which is a high density of dog population, okay? And then there's this peri-urban link population of dogs, and then you have all of these rural areas, okay? And we imagine that the rabies comes out of these dog populations into your rural areas. And if we can somehow start targeted vaccination of these linked populations, you might break this transmission cycle back and forth. Okay, so this is something we are interested in. We're trying to test that. Okay, because to achieve rabies 0 by 2030, okay, what people are doing in other countries, this for example in Malawi, is they're doing these mass vaccination drives. People bring your dogs with you. So all of these dogs are free-ranging, but they're owned in African countries. In India, we don't have this. We don't have that situation. Most of the dogs we have in India are not owned. They're free-ranging, have never been caught or given an injection or something like that. So you have to have a huge effort to go out and catch 70% of the population. I'm talking about 60 million dogs here, right? The other problem is dogs can also have a huge problem can also be a huge cause of concern for human health directly. Recently, packs of dogs went around killing children in villages of Uttar Pradesh. And as a retaliation, people then started killing dogs. So this is a lose-lose for both sides, okay? The animal welfare activists, mounted a counter campaign of this nature, right? And this sort of ends up pitting humans versus dogs. We shouldn't have that situation, okay? Because dogs are companion animals. They were domesticated. They belong with humans. We shouldn't be fighting battles with dogs on the streets. Instead, we need better laws. Unfortunately, the laws in India, okay, actually prevent you from doing anything to remove dogs from streets. The laws in India actually require dogs to be kept on the streets. Okay, so it says here that the dogs will be sterilized, vaccinated. After a necessary period, the dogs shall be released in the same place or locality from where they were captured. So this is after doing a sterilization surgery. Okay, under the mistaken belief that by sterilizing a certain proportion of the population, you will prevent or you'll reduce the growth rate of the population of dogs, and eventually the number of dogs on the streets will reduce. But ecology doesn't work like that. Biology doesn't work like that. Okay? If you remember the lines of Jeff Goldblum in Jurassic Park, nature finds its way. Okay? In India, you can't even kill a rabid dog. Okay, according to the laws. If the dog is found to have a high probability of having rabies, it would be isolated till it dies a natural death. This is a very horrible and painful death. We need to change our rules. We need to change our laws. Okay? Because we are happy to kill other animals, right, when we think that they're creating a problem. We kill lots of rats. We kill mosquitoes. We have no problem with any of those. Worldwide, these, for example, are cane toads in Australia, where they're considered an invasive species and a pest, and they're eradicated. Even what we consider cute and cuddly animals, such as foxes, are eradicated in Australia because they're an invasive species. Okay? Cats, again, in Australia, they're eradicated. But we share a very close bond with these species, so we don't want to do that. Okay? We don't want to be cruel to them and cull them indiscriminately. Okay? On the other hand, we tend to go overboard sometimes, right? And hoard animals. So this is this lady. This is the lady from Delhi, who's very famous for feeding 400 odd dogs on the streets. But it is hoarding. Okay. So then that brings us down to the question of what is compassion. Is feeding dogs on streets compassion? Okay. Or is it misplaced compassion? Are we taking responsibility for our actions when we're just putting a bowl of food out there without also ensuring that this dog is not a nuisance to somebody else, is not mistreated by somebody else, does not get run over in an accident, or does not cause an accident, does not get rabid, okay, does not have litters and litters of puppies that eventually die a very rapid death in the streets? Are we just ending, just by putting some food out there, is our compassion ending there? Okay? What is the cost of this compassion? Look, remember that we are growing food for our own consumption. Okay? That is a huge environmental cost as well. Okay? So, for example, there's one study that estimated that the ecological footprint of dogs in Japan is is the same as humans. That dogs consume about 25% of the food that is produced for human consumption. In the US, is the same thing. And a rough calculation by some colleagues said that we need about 30% extra land that is required for feeding our pet animals. Okay? Remember that there are biodiversity costs associated with that land, because you cleared forests or you cleared some kind of natural vegetation to grow that land. You displaced lots of animals, so there's no compassion for those animals. But we are only being compassionate to the animals that we see as our pets or as domestic companion animals. Okay? So this comes back to my earlier question of, will India be rabies-free by 2030? I think that if we do not fix our laws, okay, of first reducing the dog population, and if we don't come up with alternate methods of vaccination and responsible dog ownership, then we will not be free of rabies by 2030 or even 2050. Okay? The reason is that no country in the world has managed to eradicate rabies when it has a large free-ranging population of dogs on their streets. Okay? So with that, I will end my talk, take some questions, And also let you know that I don't dislike dogs. This is my new pet, Rubble, with my son. So I love dogs, but only if you take responsible ownership of them. Okay? Thanks.
Thank you for very nice and talk. I'm here.
There you
are. I have 3 questions.
Yes.
They are not questions, basically queries. One of the maps which you have shown, distribution of the incidences. And if you look at the map, particularly in Jharkhand and Maharashtra, the incidence is very, very less. So from your research, have you found any relationship of the incidences versus different parts of the country or society? That's one question. Second, in Pune city, you have shown the number of the such incidences, again, animals increasing, from 2017 to 2018. I would like to know what was your sampling strategy? I mean, because it is just 1, 2, 3, and it is going. So did you sample the entire city at one go and then kept on adding, or just randomly you have done? Third, you have tried to address this menace in the last of your lecture. Do you have any plan at a national level how to eradicate this? Any constructive idea?
Okay, great.
Thank you.
Thank you. I'll talk about Pune. So the Pune city is what we did is worked with this organization that has been for years receiving calls for rescuing animals that are in distress across the city. So there is a sampling bias, wherein as the months progressed, they basically started testing more and more different kinds of animals, even animals that they hadn't earlier suspected to be rabid. So that increase may not be an increase, but the cumulative numbers that you show up. So now that we've established that we are reaching sort of sampling saturation, that we are now sampling across the city evenly, we can be fairly confident that there was a pattern of spread from a few areas to going outwards. So that pattern seemed to be there. Sorry, can you repeat your first question again? Yes, the map. There is severe underreporting of rabies in India. So that map, by no stretch of imagination, even for Jharkhand, even for Maharashtra. So in Maharashtra, we had sampled 3 public hospitals from 3 districts where we had gotten records. And we found that Sholapur and Ahmednagar had recorded rabies deaths. Osmanabad, which is actually the poorest of these 3 districts, had 0 rabies deaths. So that's a clear case of underreporting there. It's not that they don't have, because rabies is not a notifiable disease in India yet. Okay, so there is massive underreporting. I will quickly talk about the third one, and then I'll go on to more questions. Is responsible dog ownership. That has to be your priority. That if you don't take control of dogs on streets, you're not going to be able to reduce rabies at the scale that it's required. Okay,
right,
so
we'll
that needs to be the action plan. Yes.
Sorry, we'll take one last short question.
My question is there any government policy in place for vaccination of dogs? Because the number, or has it been left to charitable organizations to do that job? Or at least a policy in place for removal of the stray dog population from the streets. Is there any policy in place?
Well, in fact, as I showed, the policy is against the removal of stray dogs from the streets. Yeah. So, and the policy is to have charitable organizations be in charge of the vaccination and ABC. This is law. This is the rules. So we need to change that.
The numbers are so huge that charitable organizations alone may not be able to do
I agree with you, absolutely, and therefore it needs to be done at a different scale. And we need lobbying against that, because currently the animal welfare lobby in India is very strong. We are talking to them, so I work with several different organizations, and they all recognize the scale of the problem. And we need to fix it. We need to fix it somehow. Currently the case is being heard in the Supreme Court of India. Whether these rules and these laws are constitutional or not, and I'm hoping in the next few months we'll have a clearer picture on how to move forward.
All right, hate to be the party pooper, but let's thank Abhi.
Thank you.
And like all rock concerts, every rock concert comes to an end. We are at the fag end of our rock concert, but before you leave the hall, some housekeeping announcements. So we have lunch for all of you. It is across in the halls Jacaranda and Silver Oaks. Jacaranda also has an exhibition of science images. Please do check that out. Make sure you wear your yellow wristbands for lunch. That's in your bag. Those of you who are heading out to Vigyan Bhavan from here, your invites, physical invites, are with your coordinators. If the coordinator doesn't have it, please come contact one of us at the registration. The college students already have their vehicles, so please finish your lunch. I mean, we always say that please finish your lunch quickly. Please do that so that you can head over. There's a protocol. Though we do not have, in general, vehicle arrangement if it's not for the colleges that have already contacted us. So, please go meet the speakers. They're here for your laugh during lunch. Let's not let them eat and let's continuously have them talk. You eat, but they should keep talking, and they should feel tired, and let's see what you can do. And thank you so much for being here. Without an amazing audience, there's no rock show. Thank you again.